Welcome back, everybody, to the second video in our immunology series. This video is going to pick up where we left off and discuss some of the properties and mechanisms that cells are going to employ to deal with infections. So, the first thing we're going to talk about is phagocytes. Now, phagocytes do a very important process called phagocytosis. Now, phagocytosis is where you have a cell that's going to ingest something. We oftentimes think of it as a foreign microbe, but it can be a piece of cellular debris, or in some cases, even eukaryotic cells like red blood cells. So there are a number of cells that can perform phagocytosis, and we refer to these cells as phagocytes. We got our neutrophils, eosinophils, dendritic cells, macrophages. Those are the major ones that will carry out this process. Why did you change slides? Don't quite get that. Okay. Now, out of all these cells, macrophages have an exceptionally important role to the host. So all phagocytic cells will respond to an infection once it occurs, and they're brought there by the presence of chemical mediators released by both damaged cells and the first cells that arrive to the area, like your basophils. But the macrophages do this process where they're already there. It's something we call immune surveillance. So when a macrophage is produced and it gets into the blood for the first time, they will travel throughout the body via the bloodstream, and eventually they leave the blood and enter into the tissues. Now, some macrophages will spend their lives going into one tissue, looking around, saying, oh, wait, no signs of infection here, and then leave and go back in the blood. And they will spend their time just going into the blood, into tissue, back into the blood for their entire life. We call these wandering macrophages, and those are the ones that will respond to an infection when they're recruited there. But other macrophages become fixed macrophages. That is, they get into the blood for the first time as a monocyte, they exit and become a fully functioning macrophage. They go into a tissue and they don't leave. They just wind up in the tissue, they look around and say, I'm staying here my whole life, all 7 days of it. And what they will do is basically patrol that tissue, looking for signs of anything wrong. Now, the vast majority of the time, they're not going to find anything wrong, and they'll die there. They'll undergo apoptosis, they'll break down, and the nutrients will be reabsorbed by the body. But if, say, you had some macrophages patrolling right underneath your skin, and you cut yourself, and then you had microbes there, those macrophages are going to do two really important things. They're first going to release chemical signals which are going to help recruit more cells to the area because they found something that needs to be dealt with. And then they're going to go charge off and go try and destroy that microbe, whatever the heck is there. So they're going to basically be an early warning system that's going to be in place. Now, one thing I do want to point out, that when they do this, they present a small problem histologically. We know about them today and what they're doing, but back in the day, when early histologists were first looking at these cells, when they saw them, they didn't recognize them as being immune cells. And so they named them. And unfortunately, what we have is a bunch of the same cell, the macrophage, with many different names. So in your liver, we call them Kupffer cells. They're microglial cells in nervous tissue. Alveolar macrophages in the lungs, and the list will go on and on. They didn't recognize that this is all the same cell that's just residing in the tissue. They thought it was tissue-specific cells. Now, all these macrophages that are flowing throughout your body and monitoring your tissues create what we call the mononuclear phagocytic system. older textbooks will call it the reticuloendothelial system. This is the macrophage immune surveillance network that every human body is going to have. And it really is an early warning system when we have microbial invasion. All right. So let's take a second and think about what's going to happen when you cut yourself. You get a cut and microbes start pouring in. You are going to see granulocytes, in particular neutrophils, dominating in the first phases. Yes, there will be a few macrophages there, those that were in fixed tissue, and a couple that would be nearby, that will be called in by the initial macrophage. However, the vast majority of cells that are going to respond are the granulocytes. And you're going to see a huge portion of those being neutrophils. Now, neutrophils are very active in phagocytosis. But most of the time, they can't clear an infection. They're highly active and they will kill many bacteria, but they're generally not able to achieve sterilization of the wound, getting rid of all the microbes. Now, if we sit there and we watch the progression of that cut over time, and we took our sample at day 1 and we saw the neutrophils dominating the immune response. If we come back at late day 3, day 4, we're going to notice that all of a sudden there's a whole lot less neutrophils present and there's a lot of macrophages. The longer you have the infection, the neutrophils get replaced by macrophages. And this is a good thing because macrophages are a superior killing machine. We have neutrophils that we throw at the infection early on, and quite frankly, we don't care if they die. Then we bring in macrophages, superior killing machines that, quite honestly, they need some help to get there. We're going to talk about activation of macrophages in a little bit. But once they're activated, man, they are just a superior killing machine to the neutrophil. They can go into a wound and sterilize it and get everything cleaned up, including cellular debris and dead immune cells. This is important because we got to get all the crap out of the way so that the healing process can happen. And that's something that macrophages are better at. So we throw neutrophils in there early, we get our macrophages all geared up and activated, and then we bring these activated macrophages in, and they clear out the infection. They will help us achieve the sterilization of that wound. So early in infection, neutrophils, later in the infection, macrophages. Now, whether you are a neutrophil or a macrophage, you do carry out pretty much the same process. The process is called phagocytosis. It's a 7-step process, okay? And yes, I would highly recommend you know these 7 steps for your next exam. Now, I like to go through the 7 steps with an image, which is why I have these guys here. So what happens is our phagocytic cell, this guy here, is going to begin by hunting down its prey. So microbes leave a chemical trail behind them. The macrophage or the neutrophil, they can detect that chemical trail and they will follow it. So this guy is going to follow this microbe, and when it gets close, it's going to extend its pseudopods out. So that's going to be an active cellular rearrangement that causes the membrane to send out these extensions. These guys are going to come around and surround that microbe. And then they're going to pull it on in. So that's what's happening. This guy is getting surrounded by the pseudopods and being pulled into the cell. But we're not going to just release it willy-nilly into the cytoplasm of the cell. As these pseudopods come around in the ingestion process, the membrane is going to surround the microbe. So it's brought inside the cell, but it's brought inside basically in a membrane-bound vesicle or a vacuole. We refer to that as a phagosome. So our microbe is inside the cell, but technically not. It's not in the cytosol. It's in this phagocytic vesicle. Now, we need to kill this microbe. That's the whole point of phagocytosing it. So what we're going to have happen is we have our lysosomes Remember, these guys are chock full of all sorts of digestive enzymes. The lysosome is going to fuse with the phagosome, which you can see happening right here. And when it fuses, all those digestive enzymes get dumped onto the microbe. This creates the phagolysosome. Those enzymes are going to go to work. And they are going to digest up that microbe from the outside in. The microbe was going to fight back, and it will try and limit as many of the digestive enzymes as it can, but it will get overwhelmed more often than not, which is going to result in the death of the microbe. Now, these enzymes will do everything they can, but they can't completely digest the microbe. So we get little pieces of indigestible material, which we refer to as the residual body. So it's still in the membrane. The enzymes have done everything they could. The macrophage will neutralize the enzymes and then discharge it. They will exocytose it out into the tissues. Now, one thing you need to understand is that when they release this out into the tissues, your tissues are bathed in fluid. That fluid begins in the blood. So it's the fluid portion of the blood that flows into the tissue, and then the tissues drain into your lymphatic system, where the lymphatic system will purify everything and dump it back in the blood. It's a circular system. That's going to become important later on. Don't forget about this material. It's going to come back when we start talking about humoral responses. All right. So I answered this in the first video, but we're going to come back to it now. So you're a phagocytic cell, and your job is to find foreign things and kill them, and cell debris, things like that. You got to destroy it. How do you know what to destroy? How do you recognize a bacteria and that a bacteria is foreign? This was a question that wasn't answered until really the late 80s, when we discovered what we called toll-like receptors. A toll-like receptor is a receptor on the membrane of our phagocytic cells. And what they do is they recognize things that are uniquely foreign. So we've talked about bacteria and viruses and some fungi. And we've compared them to, our cells. We've done that for a reason, because we highlight the differences. Well, the immune system is going to take advantage of those differences. So we don't produce peptidoglycan. We don't produce LPS or teichoic acid. We methylate our DNA differently than bacteria and viruses do. We don't have things like chitin. We don't form lipid monolayers, but the microbes do. That's the things that the toll-like receptors are going to identify. They will bind to them. We don't have it. It's not part of our genetic makeup. It's not part of our protein complement. And so if they're binding to it, well, whatever the heck is producing it clearly cannot be you, because you can't make it. And that's how they know it's foreign and how to deal and respond to it. They're recognizing these things that are uniquely not you. But it's a very non-specific thing. Every gram-negative cell produces LPS. Every gram-positive cell produces teichoic acid. It's not telling the immune system who it is that's invading you, just that it's foreign and it needs to die. So it fulfills that non-specific role that we have going on with our innate immune system. All right, so we got the process of phagocytosis. That's one way that cells are going to help deal with the infections that can occur when a breach happens. Another thing that cells can do is induce inflammation. Now, one thing I want to make absolutely clear, inflammation is not a microbe or microbiology specific response. Inflammation is a response to tissue damage. You sprain your ankle, you get inflammation. You cut yourself, you get inflammation. You get a viral infection, you get inflammation. It's not what's infecting you that's causing it so much as it's the damage that's resulting from it. It's tissue damage responses. It doesn't matter how you get that damage. If you burn yourself, you cut yourself, you freeze yourself, you tear crushing injury, it all results in the same response, inflammation. And that's because of how inflammation is brought about. Compounds released by damaged cells. Now, there are 5 cardinal signs of inflammation. We have redness, pain, heat, swelling, and loss of function. You look at these first 4. These can all be brought about by one thing, vasodilation. It's a huge part of inflammation. Vasodilation basically means your blood vessels, in particular your capillaries at the site of the injury, are going to get leaky. More blood flow to the area causes the redness. Blood from your core is hotter than blood in your extremities, which provides the heat. Excess blood flow leads to swelling, and swelling, caused by the blood, puts pressure on nerve endings, causing pain. Now, why are we so interested in bringing more blood to the area? Well, remember how microbes are flowing through your body. Immune cells are flowing to get to the microbes. They're in your circulatory system. So by increasing the blood flow to an injured area, we're increasing how many immune cells we're going to bring to that area. Now, the loss of function is brought about by many things, and pain is a big part of it, so is swelling. And that is an evolutionary trait that is stuck with us. If something is damaged, our body needs us to coddle it and to protect it and to care for it. That's where pain and loss of function come in. If you broke your ankle, for example, and it didn't hurt and it didn't swell to the point where you couldn't bend it, you'd just keep walking on it and you'd cause a lot more damage. We need to lose our function, and we need the pain there to tell us something's wrong, baby, and take care of this until it's better. We need to give the body the chance it needs to heal itself. All right. Now, lots of things induce inflammation when we start thinking about microbes. And this is where we have to differentiate something. Inflammation can be a good thing, and it can be a horrible thing. Inflammation is good when it's really short in its duration and it's intense. Inflammation is really bad when it becomes chronic inflammation. A lot of chronic disease infectious diseases will result in chronic inflammation. Chronic inflammation is almost never a good thing. It's nowhere near as intense, but because it's lasting these long periods of time, you start getting off-target effects, and it starts affecting your joints. It starts affecting your blood vessel walls and your heart, and you start to have lots of problems that develop from it. Chronic inflammation is generally not good. It's a bad thing. Acute can be very helpful, but chronic tends to be bad. All right, so why do we make an inflammatory response? Well, we want to do 3 basic things with our inflammatory response. Whatever's causing the problem, get rid of it. That's the first thing we want to do. If it's a microbe, kill it. If we can't accomplish that, say there's a microbe occurring, but we can't sterilize it out of the body, contain it, wall it off, keep it local where it is. Don't let it spread. Buy time for the adaptive immune response to get generated so that it can come deal with it. And then finally, repair. We've got to fix the tissue. If you have a cut in your finger, until you seal that cut and repair the skin, you are at risk for more microbes coming into the wound, even if you've managed to sterilize it. You gotta fix the problem. So until we get the tissue repaired, we're at risk. So we have those 3 primary functions. Kill it if you can. If you can't kill it, contain it. And seal the breach so more microbes don't come in. The way we're going to accomplish this is basically through a 3-step process. Increase blood flow and get the blood vessels to be more permeable so those immune cells can get out easy. Bring the immune cells out, get them into the tissue, kill off the microbes, and then fix the damage that has been done. So, all right, we're going to walk through those three processes. Inflammation occurs in response to injury. So say you're at home after you get done listening to this video and you're chopping up some chicken. Chicken is a dirty bird. There's lots of bacteria on chicken. Cook your chicken and watch your cross-contamination. We'll talk more about that when we get to gastrointestinal diseases. So you're cutting your chicken and you make a mistake and you go and you cut yourself with your knife. Because the chicken was dirty, your knife is dirty. And you've just breached this beautiful epidermis layer, pushing microbes into the wound. What's going to happen when this happens? Well, the damaged tissue, all the cells that haven't undergone apoptosis, cells down here, they're going to detect damage. And they're going to release a slew of chemical mediating compounds. Okay? These chemicals are going to bring about the vasodilation and the increased permeability. That they're going to do is bring more blood from the body to this area. And then the blood vessels themselves are going to become more leaky so that the fluid portion of the blood can flow out of them. This is going to start the formation of a little abscess under there. Now, we're also going to start forming a clot. We need to form a blood clot. We need to get blood to form a temporary structure that's going to cap this off. We need this breach sealed to prevent further microbes from penetrating it. So we're going to form a blood clot to get it sealed down. And we're going to start forming that abscess. We're going to fill the area up with serum. That's going to lead to some of the swelling, and you're going to start to see redness. A little bit of pain is going to start to occur. Now, if we really focus in on these blood vessels, we're going to see that in addition to leaking the serum, or the fluid portion of blood, which is plasma, not serum, excuse me, I said that wrong. The plasma is flowing out. Well, we're also going to start to notice immune cells getting out. So in response to some of those chemicals that are going to be produced, the cells that line your blood vessels, we call them endothelial cells, they're going to put little proteinaceous markers on themselves. These will only be put up near the site of the injury. And what happens is these guys will bind to our immune cells as they flow by, and they slow them down and get them to stop. Red blood cells, which are carrying oxygen, we don't care about them, well, they will just keep right on flowing by because they can't bind to these receptors. But the immune cells can. So they bind to these receptors, they stop, and then they're going to squeeze their way between the endothelial cells. So when the cells express these proteins that bind and slow down the white blood cells, we refer to that as margination. Okay, the endothelial walls near the site of the injury get sticky, and the white blood cells bind to them. Then when they squeeze between the cells, that is emigration, getting out. And once they're out, now they're in the tissue. They have all that blood plasma there with them, which is going to help nourish them during this time. They can then undergo the process of phagocytosis, find all the microbes, and destroy them. A few macrophages will be present, and a lot of neutrophils. So let's assume for a second that this response works. They are able to kill off all the microbes. We formed our blood clot. The blood clot kept us from bleeding out and provided a short-term solution to this gaping hole. Well, we will consolidate our blood clot and dry it out and form a scab. A scab is going to be more durable on the surface. And what the scab's job is to literally be a protective cap to that damaged area so that we can go through the process of regenerating the epithelium. This is a process that's going to take days. Yes, we will release things like epidermal growth factor, which will accelerate the replication rates of cells in the basal layer of our skin there, but it's still going to take days. Now, we will regenerate our tissue. And when the tissue is sufficiently regenerated, the scab itself will fall off. Scabs do not need any help from you to fall off your body. Do not pick your scabs, and advise your patients not to pick their scabs. It can lead to scarring, and if you pick it too soon, it can lead to reopening of the wound and further infection. All right. So we've got phagocytosis, we've got inflammation. There's a slew of other things that are going to happen. We're only going to talk about 4 of them here. We're going to talk about complement, interferon, transferons, and antimicrobial peptides. And we're going to focus a little more on complement than we are on the other 3, but realize the other 3 are still very important. I just can't talk about everything. So what is the complement system? Well, the complement system is a defensive protein cascade that's going to occur. All the proteins are produced by your liver and are flowing through your body and your circulatory system as we speak. They are all produced and they're all inactive. They will get activated by the immune system. Now, we'll talk about how they get activated later on. I just want you to start working on the pathway here now, because we need to learn a few things about other parts of the immune system before we can talk about their activation. Now, the whole point of producing complement is that it's going to carry out 3 antimicrobial functions. It is going to produce cytolysis, that is, it can lyse foreign cells. It is going to induce inflammation, and it is going to lead to phagocytosis. We are going to do a process called opsonization. We are going to coat our microbes in something that's going to make them easier for phagocytes to deal with. Now, the complement system is a very powerful system, which is why even when it's activated, it only stays activated for a very short period of time, and then it's shut down. Because the complement system, well, it's designed to target foreign things. Yeah, it has a habit of targeting our cells too. So we don't want the complement system on too long. It can actually lead to autoimmunity. All right. So this is the complement cascade. And yes, you should know this cascade. So it begins with complement protein C3. C3 literally says complement protein 3. That's wholly inventive naming there. So what happens is C3 is, when it's stimulated, now we have evidence from other parts of the immune system that there's an infection going on, it is stimulated and it fractures into 2 parts, C3a, C3b. C3b is what we call the binding portion. It's going to continue the cascade. C3a is an activator. Now, by itself, it can't activate, but it's going to get some help. So once C3 splits into C3a and C3b, the C3b is going to bind to complement protein 5, C5, causing it to fracture, and to C5b and C5a. Now, the C5a and the C3a are going to tag team together, go find themselves a mast cell, and stimulate that mast cell. The mast cell is going to produce immune modulating compounds like histamine and leukotrienes, prostaglandins. This is going to lead to inflammation. Okay. Now, the C5b is going to bind with C6, C7, C8, C9. Lots of C9. And it's going to create something called the membrane attack complex. So you're going to get your C5, binds to C6, binds to C7, C8, and then lots. I'm just going to repeat this one with circles. Every circle is a C9. It's going to form this circular structure. That circular structure can insert itself into the bacterial membrane, creating a pore, which results in lysis of the cell. Okay. Once C3b here is done, it's caused the fracture of C5, it's going to leave, and it's going to go find a microbe. There's our microbe. And it's going to bind to its surface, and it's going to surround it with molecules of C3b. my computer apparently wants me to go to this slide. It's changing without me touching it. It's going to do what we call opsonization. The way to think about opsonization is, well, let me tell you how I think about it. It's like when you take a strawberry and you dip it in chocolate. Why would anyone want to eat a strawberry? Oh, they're terrible fruit. And I can prove it. Name me another fruit that puts its seed on the outside. Oh, just terrible, terrible fruit. All right, all joking aside, why do we dip a strawberry in chocolate? Because it tastes better and you're more inclined to eat it. It tastes good. Well, that's what we're doing to the microbe here. Immune cells will eat microbes, sure. But if you take that microbe and dip it in chocolate, i.e. you opsonize it with molecules that that your immune cells really want to bind to and really want to ingest, you will enhance your phagocytosis. So I tend to think of opsonization as, well, you're chocolate dipping the strawberry, or you're coating the microbe so that it is easier for the phagocyte to do its job. So we enhance opsonization, or enhance phagocytosis with opsonization. We have the inflammation from the C3A and the C5A that are stimulating the mast cells to produce inflammatory molecules. And we're going to have the formation of that membrane attack complex where C5b binds to 6, 7, 8, and then we get a whole bunch of these C9s that create this ring. This is an ungated channel. That is going to allow water in. We are going to have cytoplasmic contents flowing out, and we are going to wind up with a lyse cell. So this is what complement really does. Starting with C3, going through C9, these are the major proteins. There's, like I said, over 30 of them. These are the ones I want you to focus on because they're the ones that are going to have the effects that we're looking for in the body. So that's what complement's going to do for us. 3 mechanisms for enhancing our innate immune responses. Then we have these other ones. I told you we won't talk about them as much as the last slide we're going to cover. We have interferons, transferons, and antimicrobial peptides. We'll begin with interferons. You have a viral cell, or a cell here, and you have another cell next to it, and another cell next to it. Okay. This cell in the middle is unfortunately infected with a virus. It's doomed, and it knows it. It's eventually going to have to be killed to get the virus out of it. What it's going to do is try and help its neighbors not suffer the same fate. So it's going to produce interferons. These interferons are going to be released and get sent to the neighbors. What the interferon is going to do is turn on antiviral peptides and pathways. So that when this cell succumbs to the virus and the virus is released and the virus then tries to infect this cell is now going to have some defense against it. It's going to fight back because it knows the virus is nearby. It's like raising a warning bell. You found the virus because it infected you and you're in trouble. But rather than just die without saying anything, the cell rings the alarm bell and all these other cells start turning on their antiviral processes. They're not going to stop a viral infection, but they can slow it down. Remember, one of the key things about innate immunity, buy time for adaptive immunity. So this is one of the ways that we're going to slow down our viral infections with all these interferons flowing through our cells, making sure that they are as inhospitable to a virus as possible. Transferins. Transferins are part of our body's system that keeps our iron all locked up. Remember, we talked about metabolism, and iron's a key micronutrient. It's involved in many biological pathways for both humans and bacteria and fungi. They need iron. So to try to make the human body a less hospitable place to them, we keep all our iron locked up. It's kept in hemoglobin in the blood. It's kept in ferritin in the liver and in blood, milk, saliva, tears, things like that, we have it as transferritins. The iron's all bound up in molecules, and if the organism wants it, it has to rip it out of those molecules, which they do have proteins that will allow them to do that, but it slows them down. Again, this is about slowing down the microbial growth to give our adaptive immune system the time it needs to come up. So we don't have free-flowing iron throughout our bodies. It's the function of those transferrins. And finally, we have antimicrobial peptides. These are literally short little pieces of proteins, 6 to 20 amino acids in length, that cells in our mucous membranes will produce and secrete in that mucus. They function in a very similar fashion to the membrane attack complex. They will insert themselves into the bacterial membrane and create unregulated pores, which can result in their lysis. So we're going to see those being produced as well. These guys are actually extensively studied now, because one thing we've noticed is we produce them, and the bacteria don't seem to have any defense against them. If we could somehow figure out how to exploit these and use them much like we use an antibiotic, they could help us deal with this issue we have with our antibiotics in terms of antimicrobial resistance occurring.